there, everyone, and welcome to the wonderful wild world of photosynthesis. This video is going to serve as a general review of kind of what photosynthesis is, why organisms do it, why do we care, what's the role of photosynthesis in the Rube Goldberg machine of life. And then this video is also going to kind of get a little bit more so into the nitty-gritty of the light-dependent reaction. There will be a follow-up video if you're interested in the dark reaction, and once that's finished, I'm going to link it in the description. But for now, this is just going to be what is photosynthesis and what is the light reaction? What's that first step? So, as far as kind of what photosynthesis is, we can get kind of a starting idea just looking at the word photosynthesis. So if you know anything about your roots, you probably know that the word photo means light. And then when we talk about synthesis, talk about synthesizing something, we're putting something complicated together from lots of small parts. So building. Okay, so we know we're using light and we know we're building something. But what does that actually mean? So the goal of photosynthesis is to capture light energy here. Light energy in a way that organisms can use, which in this case is like chemical bonds, kind of like we've been talking about with like glycolysis and cell metabolism and all those things in chemical that organism. All right. So how do we go about doing this? Well, not how we go about doing this, but how plants and cyanobacteria and all other photosynthetic organisms go about doing this. So just like everything else we've talked about, this is just a chemical reactions, a series of redox reactions. Redox is very, very, very important to understanding photosynthesis. So if you think that you need to review redox, you should probably do that before getting into this video because this might not make a ton of sense. But let's get into the overarching reaction that's happening here. So of course, we're going to need our products and our reactants. So when I talk about products and reactants here, we're looking at the total process. If we look at everything we put in the very beginning and everything we get out at the very end, that's what I'm talking about here. Because both parts, the light dependent and the light independent reaction, have like intermediate reactants and intermediate products, but we'll get there. So the quote unquote ingredients for photosynthesis, reactants. It shouldn't come as a surprise to you, or at least I hope it doesn't, that we're going to need light. It's in the name. No matter what kind of photosynthesis you're doing, oxygenic, anoxygenic, you're going to need light. And then of course, because our goal is to capture this light energy in chemicals that organisms can use. So we're going to need some sort of element that this energy is being like affixed to. And for photosynthesis, this is generally carbon. So we're going to need a carbon source, which is CO2. And then you're also going to need an electron donor because remember, electrons are kind of like the currency that makes all of this happen. This is where our energy kind of is. So we're gonna need an electron donor because we gotta get our electron from somewhere. And this is kind of where we see the difference between oxygenic and anoxygenic photosynthesis. It's the main difference between those two is where that electron comes from. So oxygenic, as the name suggests, you get oxygen as one of your reactants. Oxygenic. So we're going to need a compound that has oxygen in it, and for oxygenic photosynthesis, that is water. H2O. Gotta water your plants. This is one of the many reasons you must water your plants, is that they're gonna need some water. And then for anoxygenic, there's a bunch of stuff you can use. It's just not water. The most common one that you'll probably see is um H2S, so hydrogen sulfate. So this is probably one you should know. A lot of the organisms that use anoxygenic photosynthesis use this, and then water. I'll probably just call this water here too. Hopefully you can recognize H2O as water by this point. Um, so reactants. That's everything that you're going to need to input into this, into the machinery that we're going to get into in a little bit to reach this goal. But now, what do we actually get out of this process? Or not process, oh my gosh, sorry, product. So, the main product, because remember we're getting chemicals that chemicals that organisms can use. This is our goal. If you remember one thing from this, it's that that is the goal of photosynthesis, is to capture light energy and store it as chemical energy. So I'm gonna make a note. It's a chemical energy. So light energy into chemical energy. That's the big thing. Honestly, that wording is better than what I had. So light energy to chemical energy. So how are we going to store this light energy as chemical energy? Well, the main product of photosynthesis is going to be your carbohydrates. So like sugars. So when plants are making glucose, this is how that happens. That's one of the products of photosynthesis. So you're going to get sugars. Sugars are very important. This fulfills the explicit goal of photosynthesis. We want our carbs, we want our sugars. Another kind of byproduct of photosynthesis in many cases, but we're going to get into exactly when and why, is ATP. You can often get ATP out of photosynthesis directly, but also if you take the sugars and you break them down to glycolysis, you can also get ATP. But when you do cyclic, or sorry, because it really depends on what photosystems you're using. I'm getting too into the nitty-gritty. ATP sometimes. All right. So we know what we're putting in, we know kind of what we're getting out. In this video, we won't see all of these because again, this is just part one, but we're going to get to the intermediate products because remember, we have our reactants and then we're going to have another step that has some other intermediates, specifically NADPH and ATP. Honestly, I should put ATP as an intermediate product. That's a better way of thinking about it. Um, and then these two are going to go in and help us get these guys. And of course, we're going to need our carbon for that too. All right, let's get into the light reaction. Very exciting stuff. Okay, actually, one quick detour before we get into the light reaction or light dependent reaction. That's okay, that's another terminology thing that I want to clear up. Let me put all this back. Um, People have referred to these reactions in different ways. So there's two parts. Okay, there we go. Two parts. You're gonna have part one, which is your light dependent reaction. If you've been reading about photosynthesis or like watching videos on it, you might also hear this referred to as the light reaction. Also, RXN just stands for reactions. If you ever see me write that, that's what I'm talking about. And then two is light independent. Or the dark reaction. So, why do we not really call them the light and dark reaction anymore? Because it's kind of a misnomer. So, both the light dependent reaction and the light independent reaction can happen or the light reaction and the dark reaction, as they used to be called, can happen in the light, and they do happen in the light. It's just that one of them, like, inherently has to happen when light is available. And the other one doesn't have to. So it doesn't mean that it occurs in the dark. It just, I guess it hypothetically could. So light dependent reaction and light independent reaction is how they will generally be referred to in this class. I just want to define these two because just out of habit, sometimes I do call them a light and dark reaction, but I want you to know what I'm talking about. Okay, so let's get into kind of where this is happening. So let's say we are a photosynthetic plant. Here's one of our cells. We're gonna have our cell wall. You've probably encountered some of these organelles before. You're gonna have our nucleus because we're a eukaryote. We're gonna have vacuole. Throw a couple mitochondria in there. Golgi apparatus, or not Golgi apparatus. We're gonna have an ER. There's our Golgi apparatus. But what the organelle that we're going to be talking about right here is this guy. Obviously, this is not what they look like. There are some really, really, really good pictures online if you want to look at what chloroplasts actually look like. They're really interesting systems. Oh yeah, spoilers, this is the chloroplast. So we're going to zoom in on what the chloroplast looks like. So if this is our chloroplast here, I probably should change the shape a little bit. So here's our chloroplast. And inside of the chloroplast, if you zoom in real close, there are these stacks of pancakes, or at least that's kind of what they look like. And each one of these stacks is called a granum. Granum. We'll add another one here. And every one of those pancakes in the stack is called a thylakoid. And this is where the light reaction occurs. So zoom in on then I'll just do a really brief sketch, and then we can get a preview of some of the machinery we're going to be seeing in here. So this is a thylakoid, and I'm going to do a big drawing of thylakoid pretty soon. So this is where the light reaction happens. I'll make note of it here. So, or light dependent reaction. See there, I did it. Light dependent is inside the thylakoid. And then the light independent reaction just happens in the chloroplast interior. So this one happens here, and this one happens here. Draw an arrow. This one happens in here. Just kind of generally in there. On the outside of the thylakoid membrane, which is inside of the chloroplast. Okay, so I have room to draw my thylakoid. I'm gonna move this guy really quick. Oh, another thing that you will very quickly discover is that I'm a bit technologically inept, but it'll be fine. Okay. Oh my gosh, maybe it won't. There we go. All right, I'm gonna do this on a new layer because we're eventually gonna make this real big. So here's our thylakoid sac. Big flat disc. Pardon my artistry. So we're going to have our thylakoid membrane, and oh, I'll label this as thylakoid. So this whole or mini unit is a thylakoid, and then inside this space in here, all that is what we call the thylakoid lumen. So that's just the space inside. And then we're going to have a couple of really, really, really interesting, at least I think they're interesting, proteins kind of happening up in this top bit. I should probably space them out a little more. I'm gonna have to draw a lot of stuff happening with these guys. So we're gonna have 1, 2, 3, and of course this is a very simplified model. And this next one, maybe you'll recognize. I mean, of course this is a blob model, but a lot of professors I've had have represented it like this. This is our ATP synthase. If you thought you were done with ATP synthase, I'm so sorry. You're never gonna be free. She's very important. And if you're sticking with biology and you can convince yourself to like ATP synthase, it will make your life a lot easier. Okay, but these other guys, kind of weird. Reaction centers, got PS2. Unfortunately not PlayStation 2. This is in fact Photosystem 2 and Photosystem 1. And you may be like, Photosystem 2 and Photosystem 1, why is 2 before 1? That doesn't make a lot of sense. Don't let that confuse you on the test. Photosystem 2 comes before 1. It's just that they discovered Photosystem 1 first, and they were like, oh man, got this new one, and we're gonna have to go back and change all the literature. We want to like rename them in order, so we're just gonna put them out of order. So 2 and then 1. And then this other circle guy in the middle, this is the cytochrome complex. It's often represented as just like a single dot, but what you need to know is that this is actually a bunch of a bunch of little things that we're going to get into shortly. Okay. So what were our ingredients that we needed? We're going to need for photosynthesis. Here, I'm going to get rid of these other notes here just because we're going to want more space for this. Okay. Our ingredients for photosynthesis were, here, take a second, test yourself, let it stew. What do we need for photosynthesis? Well, for starters, remember photo, it's in the name. We're going to need some light. Our big, beautiful sun. We're going to need some sort of electron donor. And for the sake of this, we're going to do, okay, if water is our electron donor, what kind of photosynthesis are we doing? Oxygenic or anoxygenic? That's right, it's oxygenic. So if water is your electron donor, it's oxygenic. If it's something else, it's anoxygenic. And the other one we're going to need is carbon, but that actually doesn't come in until part two. So right now, the big things we're going to need are we're going to need our beautiful sun or some sort of light source, and then we're going to need some water. All right, so our water, something really cool is going to happen with this photosystem. So water is really stable. It doesn't like to lose or gain electrons. It has a pretty high redox potential, which means it takes a lot of, you have to have a super high redox potential to be able to oxidize water. But photosystem 2 does, in fact, have a ridiculously high redox potential, so we can steal the electrons from water, which is just kind of crazy. So it's going to snatch up this electron from water, put it in this little dot, or not little dot, it's more than a dot, called the reaction center. And we're going to get into the reaction center pretty soon. But basically, it's going to steal the electron from water, and when that happens, this water is going to kind of fall apart. And it's going to break up into two protons, and then you're also going to get an O2 minus from this. That's the oxidation state. Oh, let me just call it O minus for the sake of this. But basically, you get your protons and you get your oxygen, and then when this happens a couple times, this is going to combine into your our O2 and you're going to get O2 gas. So when people say that, oh, like plants are so cool, trees are so cool, they breathe in carbon dioxide and they breathe out oxygen. This is what they're talking about. That's where that oxygen comes from. So next time you hear someone say that, like, oh, the water is being oxidized. Pretty cool. Okay, so now we're actually going to get into this reaction center. So the reaction center is where the light gets received. So this reaction center is full of all these little pigments, and they all have different names based on the different wavelengths of light that they can absorb. And there's a big one that you kind of need to know. It's like the big pigment in photosystem 2. Oh my gosh, my voice is cracking. It's the big pigment in photosystem 2, and it's called P680. And this number 680 is associated with the wavelength of light that it absorbs. So if 680, if you know anything about your colors, 680 is red light, I believe, which is why the pigment looks green to us, because it's the red light that's being absorbed and the green that is being reflected back. So we got our P680. This is kind of the setup for this whole process. So the sun is going to do its thing, it's going to shine on down, and light is going to get absorbed into this reaction center. But it's not necessarily going to hit the P680 first. It's not, so it's going to go to this pigment and it says, no, you're not the right wavelength. And it's going to bounce around, it's going to bounce around, it's going to bounce around, it's going to get passed around all these pigments until it reaches the pigment that has the right wavelength for this. It's going to get absorbed by this electron. It's sitting really nice there. And in a lot of models, the electron is described as literally physically moving up. But what this represents is that it's going up in energy level. It has absorbed the energy from this photon that has come through. It has absorbed the light energy, and now it has a bunch of energy and is ready and raring to go. Perfect. So we have this excited electron, that's what it's called. It's full of energy, it's super excited. And it's going to move through a series of redox reactions down through the cytochrome complex. Again, it's kind of represented in a lot of drawings as being this one big sweep, but remember, it's going from it's being oxidized a little bit here. being oxidized a little bit here. It's being oxidized a little bit here and here. We're doing lots of baby steps because if you remember from the redox lessons, if you have a big, huge, steep release of energy, if you have very different redox potentials, then you're going to have a really sudden release of energy, and those can be really violent reactions, which we do not want happening in our cell. We want to keep our cells safe. So it's going to be lots and lots and lots of little baby steps. So once that happens, or so as that happens, something kind of cool goes on. Here, I need to pick a new color for this. Okay. As we pass through the cytochrome complex, if you have a environment with free protons, as this electron moves through the cytochrome complex, you're going to pump protons through. We're going to have protons moving into the thylakoid lumen. And every time that happens, you're going to get more protons, and more protons, and more protons, and more protons. This hopefully is looking familiar because this is how we form our proton gradient. Protons are being pumped from low concentration to high concentration, and this is how we get our proton motive force. Because proton gradient and proton motive force are different things, but they're very tightly associated with this. So then all of a sudden, our protons are getting a little uncomfy. They're a little too close to each other, and they wanna get out. And the only way for them to really get out is through ATP synthase. So they're gonna move through here, and it's gonna basically turn a crank to combine ADP and inorganic phosphate, that's what this PI stands for, to make ATP. Whoa, awesome! If you remember from our, oh my gosh, that's not the slide I was looking for. ATP was one of our intermediate products that we're going to need for this. Perfect. So we got one of them. But we still have this electron. Because remember, our electron, she moved. Oh, here, I probably should have used orange for this. So, moved down here. She's back here. So, the point of exciting this electron and then having it move down through the cytochrome was to pump some protons, get some ATP out of this. And now we're back in the second reaction center in photosystem 1. So it's kind of the same gig where we're going to have a bunch of pigments and then another special pigment, but it's not P680 this time. It's P700, I believe. So P700, and then I don't want to draw the sun over here, so we're going to have a second sun, which should not be the case unless you are on Tatooine. But more photons are going to hit this reaction center. It's going to bounce around, and it's going to excite this electron. And so now we have another excited electron. Hooray! So now we kind of reach a decision point a little bit. There are one of two places that this electron can go. And this is, you may be familiar with the terms cyclic and non-cyclic photophosphorylation. That is what this next step is going to be. So we're going to talk about non-cyclic first. So we have this excited electron, and you might remember our electron carriers from carbon metabolism, from glycolysis, from like the breakdown of, oh no, that's just glycolysis, or like that whole process involves electron carriers called NAD plus and NADH. In photosynthesis, we have something very similar. It's NADP plus. So there's an extra phosphorus in here. You can kind of remember that this is the one associated with photosynthesis because it has the P in it, P for photosynthesis. And this is kind of the point where this photon, this light energy, really starts to get captured in a way that organisms can use. So this electron can jump onto NADP plus, and then it becomes NADPH. Perfect. This was one of our other, oh my gosh, that's not the slide I was looking for. One of our other intermediate products. We needed NADPH. That's perfect. But remember, these electron carriers can be full. If all of the NADPH is already reduced, there's nowhere for this excited electron to go. So if the photon's hitting it, this electron's excited, but that slot is kind of taken. And this electron is sitting around like a bum, wasting wasting photons that could be exciting more electrons. It's holding up the line. So, oh yeah, so sorry. This is let me put this non-cyclic photophosphorylation. Okay, so now, sorry, I just wanted to get that down before I forgot. So instead of having this electron sitting around, taking up space, when we could be exciting more electrons, when we could be like making more things, we send it back to the cytochrome complex. Back to the cytochrome complex. Because it's excited, so it can do that, and it gets oxidized, oxidized, oxidized as it goes down in energy level. And remember, as our things pass through the cytochrome complex, they're going to pump more protons. So, and then it does that, and it gets passed back down to photosystem one. And it can get excited again, and it can keep going around and around and around and making more ATP, which is great because maybe we're not making that ultimate product that we want. We're not making NADPH, but at least we're making ATP because our cell can still use that. So that, oh yes, in this process, oh my gosh, this is cyclic photophosphorylation. All right, we're done, right? We got our NADPH, we got our ATP. Not quite. Because remember, our ultimate goal is to make carbohydrates. We're trying to make sugars. And we're not quite there yet, but we have the tools that we need. We now have the energy components that we need to put energy into those bonds and to make carbohydrates. And that is going to be part 2 of this video. Coming soon to theaters near you. Um, but thus far, I hope this was helpful. I hope this kind of cleared up how the light reaction works. If you have any questions, talk to your friends, make a study group, go to office hours, or you can email me. I'm going to put my email in the description of this video. You can also find me on Discord. I'm in the BIS2A Discord. All right. Best of luck to you all on your exam. You're going to crush it. Get some good sleep. All right. Thank you so much for watching, and I'll see you next time.